And of course, I think experiences here and during electives sort of shaped the interest in infectious diseases and the work that we did subsequently. So I think these experiences when you're younger and in training really do change the direction of your career. And so it's quite amazing to be back here speaking to clinicians actually, because all of the work we've done, scientifically in the laboratories comes from kind of clinical questions, and that's something I often have to remind myself of, because there are times when I say I don't really want to do clinical anymore, it's getting harder. But on the other hand, it's where all our research questions actually come from. So I think that so that's one of the sort of unique things that we've been able to do is to combine many different aspects of science with clinical questions. So whether it's phylogenetics or it's molecular biology, cell biology, and so forth, or increasingly immunology. So we've been able to sort of bring all of those things to bear on specific questions. That's what's made it most fun. I mean, and talks on SARS, but actually, the reason I keep talk about SARS nowadays is just because I think it was a culmination of a lot of these experiences, and so I quite like talking about it, especially to people in the HIV field, because we were working in HIV for about 12, 13 years before this all happened. And at a time when a lot of people were thinking HIV was not a problem anymore and drug resistance was not a problem. So we still kind of carried on trying to answer questions in drug resistance as they pertained to lower income settings where public health delivery of ARVs was going on. And there was very little surveillance for drug resistance or interest. And then we were able to show that non-nucleoside resistance was just really spiraling out of control in some situations. And that led to changes in policy at the end of the day. So some of these questions were there, but overlooked by a lot of the optimism that there was around HIV. So I think that having an eye on different populations is something that we're interested in. And certainly with SARS, although I'll be talking mainly about our work in the UK, what we do now is much more about looking at vaccine responses in West Africa and sort of global perspective on things. So what I thought we'd do is just talk a bit about. I mean, the talk is quite heavily based around sort of cell biology, but I think that where possible, I've tried to link it to the clinical scenarios. And the question is, why is this important? Why do we want to know about vaccine escape? Why do we want to know about cell entry? Why do we want to know about syncytial formation? And the answer is because I think these things are all linked to pathogenesis at the end of the day, and the reason we have to deal with patients in hospital. So we'll do something a bit around virus-cell interactions. An interest area of ours was chronic infections in SARS, and we'll talk a bit about that. Again, it relates to our work in HIV. And at the beginning, we were really told, and we told ourselves that as HIV scientists, we really had nothing to contribute to SARS because it was a respiratory pathogen, so the flu people should be dealing with this, and we should just sort of carry on doing our thing. But it turns out that I think SARS has much more in common with HIV than it does with influenza, for example. And then really the key thing I think that's of interest is that the rates of evolution or change of this virus is really a paradigm for what we're going to see in the future and what we've seen before. And it's an incredible opportunity to seek to understand interactions between genotype and phenotype, both in terms of replication but also immune evasion. So I think that this is another reason why we're still studying SARS, because we're able to see in real time what the variants of concern are doing, and that links directly with biological sort of parameters. And then, of course, we'll just discuss a little bit about vaccines, because that's really one of the key questions of how we're going to move forward. So it's hopefully not to, maybe everyone knows all of these things, but I'll try and sort of link these again to some relevance to clinical sort of scenarios. But if we consider ACE2 as the main receptor for SARS, now we're actually discovering that there are other co-receptors. There's a bit of debate around some of these, but ACE2 certainly is a primary receptor for SARS CoV-2, and we know that the trimeric spike protein in the virus is engaging ACE2, but it's not as simple as that, because actually to engage ACE2 and fuse at the surface, the spike protein has to be processed in a certain way, and it produces cells. So when a virus infects a cell, the spike proteins traffics to the cell surface, but during that it's cleaved by furin. And furin is a host protease that's used for processing our own proteins with maturation. And actually HIV, like HIV envelope, has to be cleaved by furin as well in order to fully mature. And so this is quite overlooked in sort of SARS biology. So the reason that cleavage event is important between S1 and S2 is that it allows them to basically to come together again once they've been cleaved. And that orientation is important because once that has happened, the following receptor engagement with base 2, temporase 2 is then able to access another site to carry out further cleavage. So if that initial cleavage doesn't happen properly, temporase 2 can't be engaged, and we'll see why that's important later on. So temporase 2 is of course the secondary kind of co-receptor that was described for SARS CoV-2. Once temporase 2 cleaves at the S2 prime site, you then get this third step, which is release of the fusion peptide into the cell membrane, and that allows the virus and host cells to kind of fuse together, and that allows the genomic material to go from the virus into the cell, and so to start the life cycle. And of course, this is again very reminiscent of HIV. HIV almost does most of these things, except CD4 is the main receptor. This is the arrangement of the spike proteins on the virus surface, and it's quite asymmetric, as you'll see. Again, there are many more spike proteins on this virion than there are. HIV has about 7 to 10, something like that. Spikes on it, trimers on the surface. SARS has a number more. But the other really important thing to note about spike is that it's a trimer, so it's 3 identical units that come together. But they can exist in these different states on the virus surface. And this is really important because they can either be open or closed in terms of the receptor binding domain being exposed up here. So we all know that the receptor binding domain is the thing that engages ACE2. And in the open form, that can happen. If it's all closed, that virus can't actually attach to a cell surface, right? So the virus has to be exposing some of its spike protein in the up position. The problem then becomes the open position is where neutralizing antibodies can then access that site. So the virus becomes vulnerable as soon as it starts exposing the receptor binding domain. So the virus is now playing a game with the host immune system, balancing infectivity with immune escape. Right, so that's the fundamental mechanism. So when we look at all these mutations on the cell surface, saying what is an escape mutation, what isn't, there's a whole other mechanism of escape immunity, which is just to hide that whole thing. Okay, so I think as we go forward in evolution, we're going to start seeing scenarios where the spike protein is closing or opening. And that's much harder to predict genotypically. There's no genotype that will tell you whether the spike is open or closed.
So what character, I suppose, what mechanisms that facilitate open and closed?
That's a big question. So these are really so interestingly, the D614G mutation, one of the first mutations to spread around the world, was actually not a RBD mutation. It was something buried in here in S2 at its position 614, and it spread very, very quickly. And we think that there are some studies to suggest that it flips the RBD open into a more open position, making it more infectious. And that correlates with this virus being much more infectious than the original Wuhan virus. So, but in terms of, there'll be other mutations that can do that, but they're more likely to, the mechanism is complex, likely involving long-distance interaction between proteins and others. So they're not easy to predict. And that's one area that we're interested in exploring. And just looking at the interface between the receptor binding domain here, or the RBM, the motif, which is a short-range region, and ACE2, you can see lots of contacts being formed. There's sort of bridges. But the reason I'm showing this is because of some of these key mutational sites are areas where neutralizing antibodies are also binding. So whilst being a critical area for the virus to gain access into a cell, it's also an area that the immune system is targeting quite strongly. And so when the virus makes a mutation to escape an antibody, it's going to affect its affinity for ACE2. So again, showing you that there's a really complicated process going on there. This is just a map, just mapping again the domains. I'll just remind you that this This is the spike protein, it's 1200 amino acids, but the N-terminal domain, which people ignore, actually most of the mutation of this we see actually in the NTD is a very plastic area. And in other there are other coronaviruses that use the NTD to enter the cell. So the NTD is actually an entry protein for certain coronaviruses. The SARS-2, of course, is the RBD that mediates cell entry, as we talked about. And then you've got these other sort of domains here in the S2 fragment. One is the fusion peptide, and it's a transmembrane domain, of course, and then a C-terminal domain. So initially, the mutation rate of SARS was estimated around 2 per month. That's quite low. It's consistent with low selection pressure, and that's consistent, of course, with the fact that there's a very short incubation period, lots of asymptomatic transmission, and the fact that the virus has proofreading. So these all contribute to low selective pressure on a virus. It doesn't really need to encounter the immune system before it's transmitted. It infects the respiratory system and then is away when the immune system has no time to do anything. So these were all reasons for the virus displaying very small, relatively modest or low mutation rates. And so we thought we had it all sorted. And of course, it fits with the paradigm of antigenic drift. This is just mutations kind of accumulating one by one slowly. The reason I'm showing you this is because pandemic viruses, such as influenza, have a method of shifting. In other words, they radically change every so often so that they can spread very rapidly. Flu does that in a very simple way because it has a segmented genome, where the genomes are divided up. And so when two different viruses infect a single cell, they can swap genetic material between the two. And then you can get a hybrid virus coming out at the end that is, to the immune system, very difficult to recognize because it's a mix now. So that's a shifting process, and that has led to some of the pandemics for flu that we've seen. And in the early days, of course, the early evolution of SARS showed you the drift, in terms of as if you sampled further in time, I could see that the distance, the genetic distance from the original Wuhan virus was sort of, it was changing in a linear fashion. If you make a phylogenetic tree, you just see different viruses, individuals kind of with very short branches. So that's what we saw in the beginning. But then, of course, with the alpha variant, we saw the group of viruses that had this very long branch and then continued to drift after that. So the big question was, how is this happening? You can see the plots here. The actual mutation rate of the variants of concern was fairly similar to the initial one, but it had kind of made a step change. And the big question was how that happened. But before we get there, this is the effect. Those mutations do have an effect. They cause antigenic shifting. This is something called antigenic cartography. It's a method developed for influenza many years ago. And Derek Smith, one of the sort of inventors or founders of that method, he's still at Cambridge and has now moved to SARS. And this is a map that basically shows you what happens when you take serum from a person infected with, say, Wuhan or D614G, and you mix it with a virus such as Omicron, and you test how well the virus, the antibodies in the serum block the virus. And the bigger the distance is, the greater the loss of neutralization. So this is basically saying that antibodies generated against this virus are not really able to combat this virus out here because it's very far away. Each of these boxes have twofold loss of sensitivity. So you can see all the early viruses are clumped together here, and Omicron BA1, the original Omicron, is out here. And actually, as they're equally important, you can see that Omicron has actually made huge jumps on its own. So many of these could be called different serotypes because there's very big distance. For example, XBB out here, very long distance between BA1. The virus is actually shifting, not just drifting, it's actually doing big shifts. You may know that XBB is a recombinant. So although it doesn't do, the virus doesn't do reassortment, it can do something called recombination, which is two different viruses can swap big segments with each other. It's very rare, and it's very rarely successful because you're introducing mutations that have not co-evolved, and so because of these long-range interactions, you can disturb those things. So it's not common to find a successful virus that was as a result of a recombination event. So how does it shift that genetically? So one option is, of course, the combination just mentioned, to make a big change to the virus. But really, again, we I think contributed some of the understanding to this by, again, a clinical scenario in hospital sort of dilemma. Somebody called saying there's a patient with COVID and we still positive after 30 days. They've had lymphoma in the past, they're on rituximab. Their antibody responses are very low. What do we do? So we sat around and said, okay, find some non-fluorescent plasma in the MDT, and so that's what happened. And then they gave remdesivir as well. That was the only drug at the time, it was 2020. And we took 23 different samples as part of the study over time, over 100 days, and then we did whole genome sequencing on the nose and throat swabs. And this figure shows you the abundance on the y-axis of different mutations in the sample by next generation sequencing. And we're just showing you the spike mutations here. But what we can see here is that after the convalescent plasma was administered, we saw a population of viruses with 2 mutations in the spike protein. And both of them have been seen in Omicron, for example. And of course, this is 2 years before Omicron emerged. Those mutations faded away as the antibodies in the plasma disappeared from the blood. Their half-life is around 2 weeks, I think, of exogenously infused antibodies. And when that happened, a different population of viruses emerged. But then when we gave a third dose of convalescent plasma from a different donor, we got re-emergence of this initial population. So you can see sort of evidence there of selective pressure driving mutational different groups of mutations. The phylogeny shows quite a big diversity of viruses, of course, and a very long branch length. Even within this patient here, we believe this virus may have been generated in a different compartment. If you look at whole genome level, not just spike proteins, this is looking at all of the virus, you can see much bigger change, many more changes between days 80 and 100. The key thing to point out here is that there are groups of mutations going up and down, which is evidence for haplotype competition, we call it. So if you have a mix of different viruses or bacteria in the body, and your plus selective pressure, or the selective pressure is changing, there will be some different, different haplotypes will have advantages depending on the environment and what's circulating. So depending on what the antibody response is or the T cell response, different viruses will be advantaged. That was changing at the time. Of course, there was a huge amount of inflammation in these patients. They were given tocilizumab towards the end of the patient died for day 100. And when that happened, you can see this huge amount of diverse noise almost. So lots of different viruses competing with each other in an inflammatory environment. So we then proceeded to figure out what the mutations were doing. We're molecular biologists, so we leveraged the fact that these were 2 mutations in different parts of the virus, one in the internal domain, one in the NS2. And we used HIV pseudotyping methods to essentially make particles of virus that had spike proteins on the outside and that were HIV on the inside. The reason we do that is because we can replace the HIV genome with a code for luciferase, which is a bright signal. And so you can measure when that virus infects the cell. And we can measure the efficiency with which different spike protein sequences are able to infect cells. We can then also mix those viruses with antibodies or serum from the patients that contain antibodies, and we can measure the ability of those antibodies to block that virus from infecting the cell. So when you do that, so we prepare viruses, and this is the Western blot just showing you protein. On that, this is P24. You can see HIV P24 is in there in those particles, but they don't have HIV on. They have spike protein on them, and we mutated the spike protein in different ways to reflect what we saw in the patients. We have observed that the deletion in the N-terminal domain actually led to a 2-fold increase in infectivity. So one of the mutations was there in the patient to enhance the virus. And this other one actually causes a defect in replication or in entry. And the answer to why that mutation was selected is escape from the antibodies and the serum that we gave, the convalescent plasma that we gave. We can measure that by taking the convalescent plasma units, mixing them with our different viruses, and measuring the degree of resistance observed of those viruses. You can see that 796 is a modest level of resistance. It's around 2 or 3-fold resistant to those antibodies. The deletion that I showed you that enhances the virus actually isn't really affected so much. In fact, it confers hypersusceptibility in some cases to the neutralizing antibodies. So you've got 2 mutations, one of which is an immune enhancer, an infective enhancer, one is an immune escape mutation. So that's the paradigm for what's happening with SARS. And then lots of other cases of chronic infection described. This one from KZN, an HIV positive individual who was chronically infected for 190 days, developed all these mutations along the way, and many of those are seen in variants of concern. So quite a lot of evidence coming out from anecdotal reports that chronic infections are really mimicking what we see in variants of concern. There isn't really another tractable way to explain why we're seeing so many variants of concern with so many different mutations, with these long branches where you've not been able to sample the intermediates. And so the most logical explanations that they come from individuals.
Ravi, the hypothesis that there could be other species.
Yes, yeah, I had a slide about zoonoses. So we know that when that there are infections in various animals, select different mutations, but I think that nobody's really shown a multiply mutated virus in any of the animal reservoirs that we've talked about. Secondly, the mutational profiles are quite different. They've got different immune selective pressures. So whilst I think it could contribute to some diversity, I don't think it's tractable. They need some variants of concern because the adaptation to a different species is very specific, right? And human adaptation, of course, is different. And many of the mutations you see that emerge, for example, that 796 mutation has a very big detriment on the virus infectivity. It cripples the virus, but it's needed for immune escape. And so when you look at those sorts of mutations, they're going to be very much species specific. The types of antibodies you make as a human versus an animal, or the effect and infectivity will be different because, of course, they have different ACE2 receptors. The sequences are a bit different, and so there are those things to consider. So just a few slides on immune evasion. Of course, many of you will know all of this, but I'd like to give us some historical context because things were much simpler than they are now when these variants emerged. In that, just starting with the alpha variant, if you recall that event, it was characterized by an RBD mutation called N501Y, and then it had actually the deletion 6970. In fact, we were one of the first groups that sort of discovered or detected the alpha variant, because when we found this mutation in our patient, we started looking at the databases, and we found this huge group of highly mutated viruses without mutation, but all these other things. So it was pretty scary to sort of see that. And then because we had set up one of the first neutralization assays, we were very quick to be able to understand whether the alpha variant was actually going to be a highly immune evasive variant or not. So we took our viruses, we coated them with spike protein from the alpha variant. We took our vaccinees, who were generally over the age of 80 in Cambridge. There were some of the first people to get the vaccine. We took their blood after dose 1 and dose 2. And we compared neutralization between the Wuhan virus and the alpha variant. And you can see there's really not much change there. So that was kind of a relief to see that the alpha variant wasn't highly immune evasive. Other people produced similar results, showing a sort of twofold effect, which was quite modest. Then, of course, we had to deal with these other 2 viruses. One that was detected in South Africa, the beta variant, and then one that was detected in Brazil. These had more mutations in the receptor binding domains. These were a bit more of a concern to people. We didn't know what was going to happen. And the neutralization profiles of these 2, of course, were worse than the alpha variant here. You can see greater degree of resistance to neutralizing antibodies for the beta variant, which is called B1.351, and the Brazil variant, P1 and P2. So we'd seen the virus characterized by N501Y, so that was seen as a core mutation, and it led to a dogma saying that variants of concern were always going to have a mutation at position 501 in the RBD. Then we saw the Delta variant, of course, which didn't have that mutation. It had different mutations in its receptor binding domain and a whole bunch of mutations in the N-terminal domain. Again, we had no idea what those were doing. What we did know is that the Delta variant here, B6107.2, was less sensitive to neutralizing antibodies. These are individuals' blood samples taken after vaccination, mixed with Delta variant spikes or spike expressing viruses. And you can see there's a drop up here. There's the Alpha variant sequence. So this was a modestly more immune-evasive virus. We had some really interesting data from India where the Delta variant emerged. Our colleagues had done a hospital study where they had vaccinated healthcare workers with AstraZeneca vaccine, and then they were doing genomic surveillance for any infections. And what was remarkable is that there was this is a phylogenetic tree of people infected after vaccine. And these are the number of days, this is the number of days after their second or first dose. So you can see it's usually about 40 days after the second dose. We got this big cluster of Delta infections. It was apparently during a coffee room, doctor's coffee morning. So one vaccinated person infected like 10 others.
We don't want to know.
Yeah. But this is at the time when people thought that after vaccine you couldn't be infected. The early days. So this was really quite shocking to many people. And CDC, I think, cited our data to inform their mask wearing policy after vaccine. I don't remember, once you had a vaccine, we thought we could all stop wearing masks. But that was short-lived. This is some of the data that informed that. And then we were able to show actually through some interesting computational methods that people vaccinated and infected with Delta variant could pass it on to other people. So you can use various methods to infer the probability that someone who's infected has transmitted it to someone else just by looking at the number of substitutions in the virus. And so we have evidence here. The thicker the arrow, the higher the probability of transmission. And the color coding is telling you how many vaccines they had. So you can see here this person had 2 doses of vaccine, but still managed to transmit the virus to another healthcare worker. So again, so some evidence for transmission despite vaccine. Now that's common knowledge, of course, and we know all that. But there was a time when we thought vaccines were going to stop everything. And nobody talks about that now. I know because of public messaging, we don't want people to think that vaccines are there to stop transmission, but actually, we did believe that was going to be the case. And then of course Omicron. So just a couple of slides on Omicron. Everyone knows this. I mean, the number of mutations across the virus genome is just huge compared to Delta, and it's spread across different regions like the RBD. Huge number of mutations in the RBD here. The N-terminal domain as well, lots of mutations compared to Delta, and then a whole bunch here in S2, which still are quite enigmatic in terms of understanding why they're there. What was interesting though is that although again, this figure just shows you data for Delta and Omicron. These are neutralization figures. So each of these dots is a person donating blood after vaccine. These are all data after second dose and then third dose. The key thing here is that Omicron titers are very low. They're kind of up in the hundreds, so not very much neutralization. The virus is escaping. If you look at Delta, much higher titers there. But then after dose 3, we found something interesting, which is that reasonable titers of over 1,000 against a virus that was nothing like the one virus. So somehow the immune system has generated a response that can neutralize the virus. Why is that? I mean, the colleagues in the US, Rockefeller, Nussensweig's group, showed evidence for quite significant affinity maturation. So in the lymph nodes, there's ongoing selection of quite diverse antibodies essentially after repeated vaccination. So the more doses you get, the broader your antibodies become. And so that's the sort of general gist of it. So suggesting that the booster was necessary to generate that breadth of neutralization. So it kind of indicated the multiple vaccine approach up to 3 doses. Whether we needed more than that, I think is more debatable, especially in the context of infection and reinfection. So just in terms of vaccine, the kind of challenges we've been facing, of course, are age, waning responses that are not necessarily age dependent, and of course immune suppression. And these are kind of the global problems we have when it comes to vaccination. Of course, there are lots of examples of this being relevant in other areas. Of course, the influenza vaccine also doesn't work so well in elderly individuals. There's a literature on that and some of the mechanisms. We collaborate with Michelle Linton, who's an expert in B-cell and aging, and she's based at the Bagram Institute. And so we did some work on our elderly individuals and showed that after the age of 80, if you looked at the first dose of Pfizer vaccine, the probability of them mounting any kind of response basically drops off after the age of 80. It's a gradual decline in the earlier years, but really after 80, something quite precipitous has happened. And the neutralization in the over 80s was much poorer, regardless of which variant you're looking at. Fortunately, of course, the second dose rescues some of that. So the neutralization titers after dose 2 now are reasonable, but they're still lower than anyone than the younger cohorts in most cases. So 2 doses recovers things in the elderly, but not to levels seen in young individuals. And of course, that's justifying the current boosting of healthy individuals. I wanted to show you one or two slides on innate immunity, because this is something that everyone ignores. Through evolution, mammals have developed anti-defenses against viruses. And of course, a new virus has to overcome those innate defenses in order to cause an infection. Coronaviruses have a whole array of, so this is just a figure to show you the sort of cascades that a viral infection will initiate. Many of them culminate in triggering type 1 interferons or IL-6, IL-1, TNF through various sensors that are sitting in your cells. We have sensors in the surface of our cell, we have sensors in the endosomes, we have sensors in the cytoplasm looking for RNA, foreign RNA. And the mitochondria are actually key in triggering and mediating some of these responses through a complex called MABs. And that triggers a whole bunch of signaling that results in interferon release. So in this diagram, I've shown some of the antagonists. So the virus, so coronaviruses have all of these ORFs that have specifically evolved, we believe, to counteract innate defenses. And then the big question was, is SARS evolving to get better and better at overcoming innate defenses? And what this paper shows from colleagues at UCL is that SARS-2, so the alpha variant, has decided to upregulate in pink genes that are known interferon antagonists, like ORF9b here and ORF6. So the Victoria virus here in blue, which is basically a Wuhan virus, has about 10-fold less expression of genes that are designed to overcome innate immunity. So the virus is stepping up its activity against the innate immune system, which I think is great to see. Well, great to very good from the book. We can wonder whether innate immunity is relevant at all, so because there's just so many defenses when we still get infected, so clearly what's innate immunity actually doing? But clearly it's doing something, because the virus has shown you that it doesn't like having to deal with it. Okay, so that's a bit on immunity. Now this sort of area that we're kind of really interested in, which is replication. Being virologists, I mean, we're not big sort of we weren't experts in binding affinity, so we did a lot of this in collaboration with others, but we do have the technology now. But to measure the interactions between two proteins, for example ACE2 and the spike protein, we can see that over time between Wuhan, Delta, and Omicron, the slope here becomes shallower. What that's telling you is that the off rate is getting longer. The proteins have on and off rates when they come together. If they bind and stay bound, then that's a slow off rate, and that's what Omicron is doing. Once the ACE2 finds its binding partner with the RBD, they stick together and they don't let go, and that increases the chance of a successful fusion event and virus infection. Again, so there's evolution going on there. How is that happening? Well, it's through contact between the two. You can see that these are known mutations, known mutated amino acids in Omicron that have been basically selected to bind ACE2 more strongly. We spent quite a lot of time developing infectivity assays in vitro to try and mimic what's happening in vivo. you can start with these Cali-3 cell lines. These are lung cell, they're cancer lines that we use in the lab. They were taken from somebody with a carcinoma. They just divide indefinitely. And when we did that with beat alpha versus delta, we found that delta was about 3 orders of magnitude better, which is a huge amount. It's 1,000 times better at replicating in those cells. We then used a more complex model to mimic the airway, which is these things called nasal epithelial cultures. So you take these are taken from donors and they've grown up. So they have mucus cells, they have epithelial cells, and air on one side and liquid on the other. So you infect from the top, mimicking airway infection, and the replication happens in here, and then the viruses end up in here. You can measure the virus. If you do that, delta again is this again orders of magnitude better than alpha, and this is suggesting that again the virus is stepping itself up each time it's involved, because it has to outcompete the virus that's already there in the population. Even more complex than that is something called an organoid system, where you take stem cells, or again donor-derived stem cells, you hit them with different cytokines and growth factors, and they start making these tube structures that mimic airways, that have alveolar cells and they have mucus cells and goblet cells. And again, if you try and affect those again, you can find that the delta was very, very much more was highly infectious compared to alpha. And so we've used those models as each variant comes along. So I've talked about binding, I've talked about replication, but how is that replication happening? It's not all about binding more spike. The viruses have also started evolving to incorporate more genomic material into the virus particles through mutations in the nucleic acid. The nucleic acid basically is a protein the virus makes to drag RNA, virus RNA, into a particle. So if you make more of the binding protein, you'll get more of the RNA in. You may know that most virus particles that you make in the cell are defective. They either don't have one or two components in them. So to get a fully formed infectious virus particle isn't so easy. It's a probability game. So mutations that increase binding would be also good news. You can see here in the paper that these mutations are associated with higher viral loads in cell culture systems. And those mutations,
So binding to what? So the nucleic acid
to viral RNA, full-length genomic RNA. Yeah. So it's 30,000 base pairs. It's a big strand of RNA, and so things can get lost, and so it needs to be tighter binding will increase the chances of a successful infectious virus particle. Tropism shifting. This is the thing we're most interested in. I think these are data from South Africa, of course. You probably will see this, where when Omicron emerged, there was an opportunity to measure hospitalization in S gene positive versus negative. Just to note that S gene positive, S gene target failure is something that probably everyone knows about. This was a marker used to detect variants. And in fact, I'm not sure if you remember that it detects the Delta 6970 that we first reported way back. So there's a sort of coincidence of things happening. And so anyone that was S gene, if you had S gene target failure, you are more likely to have a good outcome. And this was, of course, a marker of the Omicron variant, and S gene positive, in other words, the Delta variant was associated with a higher hospitalization rate. So those are some of the early stages that were replicated in other populations. It mirrored what happens in animals if you infect animals with Delta versus Omicron. These are severity scores in Syrian hamsters from colleagues in Japan. And Delta is in orange, much higher severity in hamster lungs compared to Omicron in green. So human and animal data correlated. So we started to try to understand why that's happening. We used our culture system with nasal epithelial cells. If you compare Delta and Omicron, in those nasal, they're mimicking the upper airway. Got very little difference between the two, so they look like they're replicating as well as each other. But when we used our organoids that represented lower airways, or Cali-3 lung cells, we've got quite a big difference between the alpha and delta, sort of 3 orders of magnitude. So Omicron was quite severely impaired in those lower airway organoid models. And then so the big question was why? first of all, we thought, okay, this correlates with severity. Can we take that further? And then to understand what was going on there, we had to start digging a bit further into virus entry. I told you the reason I mentioned TMPRSS2 and cleavage earlier is because for SARS to use TMPRSS2, it has to, we believe it needs to have cleaved its spike protein and produce itself properly. And then it can access this plasma membrane route of entry. And this is we believe this is the dominant mode of entry in the lungs. Okay, so for the lung infection to happen, we believe that you need to engage TMPRSS2. If you don't engage TMPRSS2 because you haven't cleaved the spike or for whatever other reason, the virus then goes through the endosomal route. In other words, it gets swallowed up into these vesicles in the cell where you have cathepsin, and that cleaves the spike protein instead of TMPRSS2. The interesting thing is that some of those innate immune sensors are based around in here. So the TLR, toll-like receptors, are very heavy, and in particular IFA10s are located here. That's why we believe that viruses have chosen to go through this route, which is to avoid innate immunity. HIV also, we believe, prefers this route as opposed to the endosome. So two routes. We hypothesized that Omicron was going down this way instead of this way, and that correlated with the lung infection disparity. And so to test that, we used drugs that block one or the other pathway. If you block surface entry with this drug, Camstat, which was actually developed as an anti-coronavirus drug, you can completely inhibit Delta, reach 100%, but you can't inhibit Omicron. Telling us that Omicron may be going through the endosome. So we test that by blocking the endosome through this drug E64. And if you this does result in complete blockade of Omicron, but not Delta. So telling us that the viruses have switched entry pathway. So I think this is really what's kept us in coronavirus research, which is that you've got reduced severity. You can see it in the animals. You can see that lung cells are less susceptible to Omicron. The upper airway cells are equally susceptible. So the lower airway replication correlates, difference correlates with severity. And we believe it's TMPRSS2 mediated. So this is really evidence that in real time, within our lifetime, we can watch the virus go through being a lower airway disease to an upper airway disease, the classical paradigm for virus evolution, which is to become an upper airway transmitter, because it's more infectious and more effective. So that's what I think is incredibly interesting. But it's a problem for sterilizing immunity because, of course, you need really high levels of local protection in the upper airway in order to prevent infection and transmission. So whilst being interesting biologically, it's now a huge problem for us in terms of transmission. This doesn't quite end there because syncytia formation is also really quite markedly different. So you may or may not know that many virus glycoproteins or spike type proteins, HIV, for example, can mediate cell fusion. So these are macrophages. If you infect them with HIV, the macrophages start sticking together to form these multinuclear giant cells. Of course, tuberculosis and TB as well. And so one interesting thing I'd like to understand more in the future is is there a parallel between what the virus is doing to make these syncytia and what the body is doing to generate these and then granulomas against intracellular pathogens. But the result of that happening is that innate immunity is activated. So when two cells fuse together, often due to a virus-mediated process, you get triggering of IRM3 and interferon-stimulated genes. This probably ends up at a systemic level, causing systemic inflammation. So there's a theoretical link between a virus that makes syncytia and uncontrolled activation and inflammation. Because if this happens in the lung, you might imagine that's bad news. And of course, autopsies from people who have died from COVID-19 show these big, multinucleated syncytia. So this is a paper in Nature with lots of some examples of this. So for some reason, severe COVID-19 pneumonitis is associated with syncytia. So then, is there a connection between fusing cells together and different variants of concern?
Maybe those are epithelial cells.
Those are typed alveolar cells, so they've done the staining. These are cells that are viral nucleic acid positive, I think that's the thing, but they're alveolar cells based on markers. And what was fascinating is that when we developed in collaboration with colleagues at the LMB, we developed a fusion assay, so we can detect when a virus has caused two cells to fuse together. So we use standard cell lines in the lab. We just make the spike protein. We don't give them a virus. We just make the cells make spike protein, and the other cell on the other side has ACE2, so they can bind together. And when they fuse, the thing goes green. So you can see Wuhan virus makes nice complement, lots of cells joined together. When we did this with Omicron spike, we saw very, very few fused cells for some reason. And when we do a time course, we can see that wild type Wuhan makes syncytia, joins cells together. Delta does it even better, but Omicron really is unable to do it. It does it eventually at later time points, but really, really slow at fusing cells together. So that's another potential reason why pathogenicity of Omicron was lower than Delta, for example. It's reduced ability to fuse cells and trigger that inflammatory process. So, this is just a slide as to say, based on all of that, where are we going now? Because people will ask, well, I've just spoken about, what things like Omicron. I guess we're seeing with Omicron evolution further immune evasion, conversion evolution. In other words, the same mutations happening in different lineages. The virus is having to encounter the same barriers in different hosts around the world. And so they're selecting the same mutations. We've seen the first successful recombinant, which is XBB. That's quite a big deal. The viruses now have to do weird things to try and overcome population level immunity. And of course, as ever, the virus is trying to balance immune evasion, but still maintaining its ability to get inside a cell. And it's provided lots of interesting insights. So just to summarize, we've hopefully shown that antigenically shifted viruses are probably arising from chronic infections in individuals. HIV, we think, is a significant risk factor, possibly relating to the fact that some of these epochs have emerged in South Africa, although we need to do more work to really understand that further. The shifted biology of Omicron BA1 towards reduced lung disease, I think, was a major step for our understanding, but also for the direction of the pandemic. The big questions we're asking now is whether Omicron is going to start evolving to reverse some of those pathogenicity changes. Will it start fusing cells together better? Will it start infecting lung cells? Now that we have a set of assays developed around, many groups have these assays, so we can, when new variants come along, we can test, whether they're doing these things and can be expected to cause more severe disease. What are we going to be doing in the future? We want to know what the limits of spike evolution are. But we may have to wait for nature to do this for us because, as I said, genotype prediction is not perfect. Allosteric interactions are critical in that because mutations far apart can actually influence each other and the overall biology. And then I guess there are a whole bunch of questions about vaccination, mucosal vaccines versus injectables. There are some reports that some of the inhaled vaccines are really good at inducing IgA, but they're not so good at inducing germinal center maturation in your lymph nodes. And so therefore they may not deal with future variants so well because you're getting a very localized specific response to a certain variant, and maybe that's not what you need going forward when you're trying to protect against future variants. So there's loads of interesting science to be done. I'd like to end by just acknowledging the guys at the lab who've done responsible for the work that I've shown, and the colleagues around the world during a really challenging time. And thank you for your attention.